Okay, welcome back, everyone. Dr. Lindner. This is Anatomy and Physiology 1. In this short lecture, we are going to talk about the cell. So let's take a look at this picture. I'm going to go over some of the terms just to show you different terms that you could be familiar with them. And then I'm going to get into the description to each of them and some basic function. Okay. Let's see if we can open this up a little bit more so everyone can see it. Great. Okay, so this is what we're looking at. If you remember back in one of the earlier lectures that I did, we talked about the organization of life, right? Where we said we had chemistry and then chemicals come together and make cells, then cells come together and make tissues. And then tissues make organs, organs make systems, and then systems make the organism. So here we're looking at the cell. And some of the main parts of the cell that I want you to get comfortable with, if you look here, let me get my pen here. I'm going to make it red so we can see that a little bit better. So right here on the outside, going around the entire perimeter, that is referred to as the cell membrane. Sometimes it's called, as you see here, the plasma membrane. Okay, so same thing, interchangeable terms, but the cell membrane is a phospholipid bilayer. Phospholipid bilayer. What we mean by that, when I say bilayer, it means two, right? So it's two layers. There's a phosphate portion and there's a lipid portion. So a phospholipid bilayer. It's like this. Let me see if I could draw it for you. So we're going to have, let's say, like this. And again, I'm going to exaggerate it just for simplicity. But under a microscope, when you look at something like this, it's going to have two layers to it. That's a phospholipid bilayer. Now, if I take this section of it right here and I blow it up, right, make it larger, we're going to see something that looks like this, phospholipid bilayer. So here, I'm going to draw these little circles like this and this. And on this side of the membrane, I'm going to draw circles here and here, which would look like this: circle, 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 circle. Those are the phosphate portion of the cell membrane, right? I said it's a phospholipid bilayer. So the phosphate portion is on the outer part of the cell membrane, like this, and it's on the inner part of the cell membrane, which is here. And they like water. So they say it's hydrophilic. Hydrophilic. Philic comes from the Greek word philos, like philosophy, philos means friend. So we're looking at something that really likes water. And outside of the cell, there's liquid, there's water. And then inside of the cell, there's liquid or water. So the outer part and the inner part is hydrophilic because it's got these phosphates. Fantastic. On the inside of the cell membrane, here is where we have fat or lipids. And it's going to look like this. The lipids look like these tails. I'm going to draw one here, a straight line, and I'm going to make a bent line. On this side too, a straight line and a bent line. The straight line, straight means it's saturated. And when you have something that has like a bend to it, like it isn't straight, it has this kink, we say it's unsaturated. So we have saturated fats like this, nice and straight. And then we have an unsaturated, which has a kink to it. It has a bend. So if you look, each of these has a straight one and it has a bent tail. The bent is an unsaturated fat. Saturated is nice and straight. So again, the cell membrane is a plasma membrane or a cell wall. That outer layer has two layers, phospholipid. The outer and the inner part is hydrophilic. That's where you find the phosphates, the spheres. And then in the middle are these lipids. One is saturated and one has a bend to it, unsaturated fat. The saturated is rigid. It gives the cell wall or cell membrane its structure, but then the kink gives it this bend, which is needed for flexibility, right? When you have a bend to it, it allows the cell wall to be flexible and more fluid-like. So you have a balance between rigidness, structure, and then part of it, what gives it its flexibility. So there's the beauty of the cell membrane. Okay, and you'll go over that a little bit more in lecture. Okay, what else do we have here? Now, outside of the cell, if we look outside of the cell wall or cell membrane out here, everywhere out here, they use the word extracellular fluid. Extracellular means outside of the cell. When you hear the term intracellular, we're talking about in here, intracellular. So there's extracellular fluid and intracellular fluid. Extracellular fluid is outside of the cell. Intracellular fluid is within the cell. Now, of course, they have other names. And you'll see that the author of the book sometimes refers to extracellular fluid as interstitial fluid. Interstitial fluid. Don't worry about the spelling for now. You can catch up with that later. The inside of the cell, which is the intracellular fluid, is sometimes referred to as the cytosol, cytosol, which we see labeled here, cytosol. Okay, it's important to know that. Extracellular fluid, intracellular fluid. Extracellular fluid is the interstitial fluid, and the intracellular fluid is called the cytosol. Okay. What else do we have? What other structures that I'll go over in terms of their function that it's important to know? Let's take a look at the nucleus. Here's the nucleus. We see it pointing to not the peach colored thing here in the center, but it's pointing to the translucent sphere, right? It looks like that clear translucent layer of that ball. That's the nucleus. And that's important because the other structure that I circled here, let me change the color for you, see if I can make it a little bit easier for you to see. Let's make it blue. How about that? Let's make this one blue. This one here that I'm circling blue that looks like that orange peach pit in the middle, that's the nucleolus. So don't confuse nucleus with nucleolus. I often see that mistake on the exam. They're not interchangeable. So again, nucleus is the clear part, and the nucleolus is the blue sphere that I circled here. Now, what surrounds the nucleus is this white structure that has these little permeations, these little holes in it. This white structure here, the white part, is called the nuclear envelope. That's called the nuclear envelope. And the little holes within it, like here and here and here, those little holes are called nuclear pores. Nuclear pores. Where are the nuclear pores? Well, the nuclear pores are within the nuclear envelope. What is the nuclear envelope? Well, it surrounds the nucleus. And the nucleus surrounds the nucleolus. Lots of terms that, you know, they're like tongue twisters, okay? Okay. So the reason that we have these nuclear pores is the nucleolus that lies deep within is going to produce something. And what it produces is going to find a way out to escape through these nuclear pores. So that's why we have them. Okay. Now, what surrounds that nuclear envelope, we see this aqua color structure, right? This aqua color folding envelope that we see here. This is called the endoplasmic reticulum. You can see the term here. I'm going to underline it in blue. Endoplasmic reticulum, endoplasmic reticulum. And you see that there's a smooth endoplasmic reticulum and a rough endoplasmic reticulum. What makes one smooth? What makes it rough? Well, you see the white speckles here? There's a white speckle and there's a white speckle. You see these like salty, grainy, rough structures on the aqua. Those are called ribosomes. Those are called ribosomes. And you see that here they're called fixed ribosomes, meaning they're fixated to the endoplasmic reticulum. They're stuck to it. Now, there's also free ribosomes. Free ribosomes are these here, they are the white speckled dots that we see in the cytosol that are not adhered to the endoplasmic reticulum. So we have ribosomes. Ribosomes can be attached to the endoplasmic reticulum, and if they are, they're called fixed ribosomes. And it makes the endoplasmic reticulum look rough. So it's called the rough endoplasmic reticulum. If we don't see those fixed ribosomes, like here, this section here looks very smooth. So we call that the smooth endoplasmic reticulum. And like anything else in anatomy and physiology, if they look different structurally, they're going to function different because structure determines function. So we'll go over each of these, what their function is in just a moment. I just want to show you right now where these are. So we know ribosomes. We know that they can be free or fixed. Free is they're just hanging in the cytoplasm or the cytosol, and fixed is when they're actually attached to the endoplasmic reticulum. Okay, we have these structures here. Everyone loves these. These are called the mitochondria. The orange circles. These are all mitochondria. Then we have these two vessels. One looks gray and one looks yellow. The yellow one is larger than the gray one, and they sound a little bit alike. The yellow one is called the lysosome, and the gray one is called peroxisome. They look different. They're going to function different, and I'll tell you what their functions are. And you should know how to identify them, and you should know what their functions are, because guaranteed you're going to see it not just on your lab, but when I was teaching the lecture portion, I make sure I put that on my lecture exam as like a matching section or something. So I would put the definition, and then you would know which organelle, which one of these organelles performs that function. Okay, few more structures to cover. Right here, we see this black circle with these yellow rods in them, okay? So the black circle is called a centrosome, and then these yellow spindles that are located within it are called centrioles. We have the centrosome, which is the black round circle, and then we have the centrioles, which are these yellow spindle fibers that are located within it. When we watch the video on cell mitosis, which means cellular division, you'll see these come into play. we have a structure called the Golgi complex or the Golgi apparatus. You see it on the top left here, the Golgi apparatus or Golgi complex. The Golgi apparatus I'll talk about in a minute, but just be familiar with what it looks like here. I'll go over their functions in a second. Okay, I also want you to see this structure here on the, it's around 11 o'clock, right? If you look at the 11 o'clock slide, we see this structure here, and we notice that here's the cell membrane, here and here in blue, and this structure, what it did, it was a vessel. It was a vesicle like this. And this Golgi apparatus pushed out a package, a vesicle that looked like this. And within the vesicle was a protein. It packaged a protein that was sealed in this round package called a vesicle. And it said, hey, I'm going to put a protein here within you, and we're going to move to the cell membrane. We're going to open up, and maybe those proteins are going to be used to recycle some of the damaged proteins within the cell membrane wall. Because the cell membrane wall is taking a lot of hits. It's getting hit by so many different things, it can become damaged. So we have to recycle and regenerate and repair that cell membrane. So sometimes within the Golgi apparatus, one of these vessels can migrate and move to the cell wall. It opens up, and the proteins within it are used to recycle, regenerate, refresh, or renew the proteins contained within the cell membrane. That's a possibility. But sometimes this Golgi apparatus makes a protein within it, and when you do proteins in lecture, you will cover what some of the functions of proteins are. Sometimes the proteins could be a hormone. The proteins could be a neurotransmitter, which is just a chemical messenger. It could be an enzyme. And sometimes the protein that is packaged within the cell has to be shipped outside of the cell. So if this vessel moves to the cell membrane wall, in this particular case, it looks like it's opening up, and whatever protein was contained within it is going to move outside of the cell. It pushes it out, and that method is called exocytosis. Exo, like exit, exocytosis. It's releasing whatever contents are within the cell. It moves it outside of the cell. That is called exocytosis. You're going to hear that term throughout anatomy and physiology, exocytosis. Okay, let's see if I covered everything here. Looks like we covered mostly everything. So now let's go through what some of the functions of the some of the organelles contained within. So I'm going to pause here, and I'm going to create a separate video on just the functions.